Welcome to Peptide-Proof, your source for rigorous, independent, evidence-based peptide intelligence. Is this the peptide ingredient that could rebuild your skin matrix? Let's jump straight into this explainer and look at the exact science behind it. Most peptide skincare conversations usually stop at the big three, where a peptide triggers maybe one or two specific proteins. But what we're looking at here is something entirely different. Palmitoyl Tripeptide-38 doesn't just do one job. Think of it less like a specialized worker and more like a general contractor for your skin. It actually orchestrates a highly coordinated rebuild of 6 different structural components all at once. Let's break down exactly how it pulls this off. To understand how it works, we really have to look at its molecular anatomy. This molecule has 2 critical parts. First is the core signal. It's a short 3 amino acid chain made of lysine, valine, and lysine. This specific sequence perfectly mimics a naturally degraded fragment of type 1 collagen. When your fibroblasts detect this fragment, they get a very clear signal. The extracellular matrix is damaged, and it's time to rebuild. It's basically your body's own natural maintenance loop, repurposed as a cosmetic ingredient. But here's the catch. On its own, this tripeptide is way too water-soluble and too large to actually penetrate the skin. And that brings us to the second part, the palmitoyl tail. This 16-carbon fatty acid chain makes the entire molecule lipophilic. That means it can easily dissolve right into your skin's natural lipids and travel deep down into the dermis, which is where those fibroblasts actually live.
And this brilliantly illustrates what happens once that signal reaches the fibroblasts. While other popular peptides primarily target collagen 1 and 3, Palmitoyl Tripeptide-38 activates an entire suite of structural proteins. First, it triggers collagen type I, generating those thick, rope-like fibrils that give your skin its core tensile strength. Next is collagen type 3. That's the finer, more elastic collagen that gives youthful skin its bounce. Third, it stimulates collagen type 4, which is unique because it builds the sheet-like basement membrane separating your epidermis from your dermis. Moving on, we have fibronectin. This acts as the crucial scaffolding to make sure all those new collagen bundles are properly aligned. Then it triggers hyaluronic acid synthesis, deeply hydrating and plumping the dermis by binding water. And finally, laminin 5. These are essentially microscopic rivets that anchor your epidermis to the dermis to prevent sagging. So that's 6 proteins, 6 different structural roles, all stimulated by a single signal.
Section 1. Beating the skin barrier. The biological mechanism is incredibly powerful, but honestly, none of it matters if the ingredient just sits on top of your face. Every topical peptide faces a pretty massive hurdle, the stratum corneum. It's basically a brick wall of dead skin cells and lipid mortar. In skincare, there's a well-known guideline called the 500 Dalton rule, which states that anything larger than 500 Daltons can't easily pass through that outer barrier. Well, Palmitoyl Tripeptide-38 weighs in at roughly 650 Daltons, putting it at a distinct disadvantage. The secret here lies in lipid partitioning. Because of that palmitoyl tail we talked about earlier, the molecule becomes lipophilic enough to slide right through the intercellular lipids, completely bypassing the 500 Dalton rule. Now, admittedly, the estimated penetration efficiency is pretty low. Only about 5 to 15% of the product actually gets through. But the crucial point is fibroblast activation. The concentration needed to trigger these fibroblasts is extremely minute. Even with low penetration, reaching the dermis at a tiny catalytic dose of just 3 to 5 parts per million is more than enough to activate the receptors. It doesn't need to pile up, it just needs to deliver the message.
Section 2, the clinical evidence. We know the theory and we've seen the delivery mechanics. Now let's move from molecular theory to documented clinical outcomes.
These are the exact metrics from independent studies. Keep in mind that in published literature, this peptide is almost always tested as part of a combination formula, but the data paints a really consistent picture. In a 56-day study by Lintner using a 5 parts per million concentration, they observed a statistically significant 9% decrease in skin redness, alongside an 8 to 9% reduction in measured skin roughness. Meanwhile, Sedoma's own 48-hour in vitro data shows a massive 124% increase in hyaluronic acid synthesis at just 3 parts per million. And in a 28-day study by Sans, 71% of subjects experienced clinically relevant anti-wrinkle effects, with visible improvements noted in just 7 days. That rapid 1 to 4 week timeline makes perfect sense for a signaling peptide that quickly turns on your existing cellular machinery.
Beyond the headlines.
Into the research. Peptide-proof.
Section 3. Formulation and stability. Now, here's where things get a bit tricky. There's often a massive gap between clinical theory and the product you actually buy off the shelf.
Experienced formulators know exactly what ingredient labels hide, and there are 2 critical pitfalls you need to avoid here. First is the concentration illusion. A brand might proudly claim 1% Palmitoyl Tripeptide-38 right on the bottle, but the raw material is usually supplied as a premixed solution containing only 100 parts per million of the active peptide. So 1% of that solution means the final product only delivers 1 part per million to your skin. That is well below the 3 to 5 parts per million clinical threshold we just established. Second is the pH window. Palmitoyl Tripeptide-38 is only stable between a pH of 4.5 and 6.5. If you layer this peptide immediately with a harsh exfoliating AHA or BHA acid, which typically sit at a pH of 3.5 or lower, the amide bonds in the peptide hydrolyze. The molecule literally breaks apart before it can even penetrate your skin.
Section 4. Stacking your routine. Understanding stability naturally brings us to the next logical step: how to practically and safely integrate this specific ingredient alongside your other powerful skincare actives.
Let's map out how this builds into a routine. If you're using Matrixyl pentapeptide-4, it's a perfect complement. Pentapeptide-4 provides a targeted collagen trigger, while tripeptide-38 delivers a broad matrix rebuild. And the best part is they don't compete for the same cellular receptors. Next up is arginine. This is a brilliant structural and dynamic pairing. Tripeptide-38 rebuilds the dermis from within, while arginine relaxes the muscle folding at the surface that actually causes expression lines. Niacinamide is another fantastic partner. It provides the vital energy support, specifically cellular NAD, required to meet the massive energetic demands of synthesizing all these new proteins. Finally, a quick note of caution. If you're using high-dose GHK-Cu copper peptides, you really should alternate days. Both of these are highly stimulatory signals, and it's best to avoid overloading your fibroblast receptors all at once. So what happens if you stop using it? I love this cellular kinetics graph because it reveals a truth that just isn't talked about enough in skincare. Because dermal collagen has a very slow turnover rate, the structural benefits you gain will actually persist for quite a while, even after you discontinue use. But look at the hyaluronic acid and cellular hydration. Those levels regress rapidly within just a few weeks of stopping the product. This proves a vital concept. Palmitoyl Tripeptide-38 is a maintenance signal, not a permanent modification. To sustain the full, comprehensive cosmetic results, you need consistent, ongoing cellular signaling. Section 5, the executive summary. We've covered the molecular structure, the delivery mechanisms, and the real-world formulation challenges. Let's bring this explainer to a close with the core takeaways. If you remember nothing else from this, quickly review these 3 pillars. Number 1, broad-spectrum signaling. Unlike isolated collagen boosters, it acts as a general contractor, triggering the comprehensive synthesis of 6 vital dermal and epidermal components. Number 2, catalytic efficiency. Thanks to its lipid tail, the minute fractions that do penetrate the skin barrier are fully capable of activating your cellular machinery without needing to accumulate. And number 3, smart formulation. Its efficacy relies entirely on proper pH environments, between 4.5 and 6.5, and ensuring the brand is actually using a true active concentration above 3 parts per million, not just a heavily diluted solution. That's all for this episode. Comment peptide and I'll send you the full peptide research summary. Thanks for watching, and we'll see you next time.